Could a single gene difference be the key to understanding why we survived, while Neanderthals and Denisovans vanished? A fascinating new study suggests it might, focusing on a protein crucial for DNA synthesis. This protein, called ADSL, exists in a slightly different form in modern humans compared to our extinct relatives. And get this, experiments with genetically modified mice hint that this tiny difference could have had a major impact on behavior. But before we jump to conclusions about human evolution, let's dive into the details. Researchers at the Okinawa Institute of Science and Technology in Japan, led by Xiangchun Zhu and Nobel laureate Svante Pääbo, zeroed in on ADSL because it's one of the few enzymes that underwent evolutionary changes in the lineage leading to modern humans. ADSL is vital because it helps create purine, a fundamental building block of DNA and other essential molecules. Think of it as a crucial component in the recipe for life itself. Now here's where it gets interesting. The ADSL protein is made up of 484 amino acids. The version found in almost all modern humans differs from the Neanderthal and Denisovan version by just one amino acid. Specifically, at position 429, modern humans have valine, while our extinct cousins had alanine. This seemingly insignificant change likely occurred after we branched off from the Neanderthal-Denisovan lineage. To investigate the potential effects of this mutation, the researchers turned to mice. They genetically modified mice to express the modern human version of ADSL. The results, published in the journal PNAS, were intriguing. They found that the modern human variant led to higher levels of the chemicals that ADSL normally processes in several organs, particularly the brain. This supports the idea that the modern human version of ADSL is less active than the version found in Neanderthals and Denisovans. And this is the part most people miss. The behavioral changes were observed primarily in female mice. In experiments where mice learned to associate lights or sounds with a water reward, female mice with the human-like ADSL variant were better at getting the water than their counterparts without the variant. This suggests that the human-like variant might have enhanced their learning abilities or increased their motivation to seek out the water. But why only females? It's unclear why only female mice seem to gain a competitive advantage, said researcher Izumi Fukunaga. Behavior is complex. This raises a lot of questions about sex-specific genetic expression and its role in evolution. Statistical analysis of Neanderthal, Denisovan, and modern human DNA further supports the idea that the ADSL mutation provided some evolutionary advantage. Mutations in the ADSL gene appeared in modern human genomes at higher rates than would be expected by random chance. This suggests that natural selection favored this particular genetic change. But here's where it gets controversial. Prior research has shown that ADSL deficiency in modern humans can lead to intellectual disability, speech and language impairment, and other problems. So, how could a less active version of ADSL be beneficial? The researchers suggest that modern humans may have had to strike a delicate balance between the potential benefits of reduced ADSL activity and the risks of ADSL deficiency. It's a classic example of evolutionary trade-offs. However, not everyone is convinced that this study provides direct insights into the success of modern humans or the demise of Neanderthals and Denisovans. Paleoanthropologist Mark Collard, who was not involved in the research, believes that the results in mice don't say too much about human evolution at this stage. He does, however, acknowledge that using mice to study the behavioral effects of genetic differences is a promising approach for investigating the evolution of our brain and behavior. Ultimately, the implications of this study are still unclear. As the researchers themselves acknowledge, the neural circuits of mice are vastly different from those of humans. More research is needed to fully understand the role of ADSL in human evolution. Future studies could investigate the specific mechanisms by which changes in ADSL activity influence behavior and explore how multiple genetic changes might interact to shape our species. What do you think? Could a single amino acid difference really have played a significant role in the story of human evolution? And if so, what does that say about the power of seemingly small genetic changes? Share your thoughts in the comments below.